Good morning guys, welcome to my YouTube channel. I'm Dr. Shachita Verma and I'm back with another video on the topic KREB cycle. So let's get started. So KREB cycle is commonly known as tricarboxylic acid cycle. Now why the name KREB? KREB after the name of its discoverer, that is Hans KREB. So this cycle, this KREB cycle is named after the person who discovered the cycle, and the person is Hans KREB. Hence the name KREB cycle. Now KREB cycle, it occurs in the mitochondria of the cell. Now as we know that glycolysis is a process, it's an anaerobic process and it occurs in the cytoplasm of the cell. But then all other reactions in the respiration, it occurs in the mitochondria of the cell. Now why the name is TCA cycle, that is the tricarboxylic acid cycle? The reason is the very first compound that is formed is citric acid or citrate. It is a 6-carbon compound and this compound has 3. Tri stands for 3. So this compound has 3 carboxylic acid groups. That is the reason this KREB cycle is also known as TCA cycle or tricarboxylic acid cycle. Now we'll start from glycolysis. What happens in glycolysis? From one molecule of glucose, we get 2 molecules of pyruvic acid. But here, when we are studying the KREB cycle, I am going to discuss about one pyruvic acid molecule first. And then, whatever energy is being produced from this one pyruvic acid molecule, we are going to double it. Why? Because one glucose molecule, in the glycolysis, it splits into 2 pyruvic acid molecules. So now one of this pyruvic acid molecule I have written over here, and from here we are going to discuss about how it goes to form oxaloacetic acid. Fine. Now let's start. Pyruvic acid is first of all converted to acetyl coenzyme A. Now here one carbon dioxide molecule is released. So it's a decarboxylation reaction. Also one coenzyme molecule, coenzyme A, is being added. Hence what is being formed is acetyl coenzyme A. Fine. Now this pyruvic acid molecule is a 3-carbon compound. It's a 3-carbon molecule. But acetyl coenzyme A is a 2-carbon molecule. Another reaction which takes place over here is the reduction of NAD into NADH2. So now here energy is produced in the form of NADH2, which further gets reduced to ATP, right? Now acetyl coenzyme A is a 2-carbon molecule and it adds up to oxaloacetic acid. This oxaloacetic acid is a 4-carbon molecule, right? Now this acetyl coenzyme A, when adds up to oxaloacetic acid, it gives us citrate. Now citrate is again a 6-carbon compound. Now here the enzyme, because what is going to happen here? Citrate or citric acid is being synthesized. So because it's a synthesis reaction, the enzyme that will work over here is citrate synthetase. From synthesis, the name of the enzyme will be citrate synthetase. Now this citrate will again gets converted into its isomer, that is isocitrate. So now iso suggests that it is the isomeric form of citrate or citric acid. So again, it is a 6-carbon compound, and the enzyme which is acting here is isomerase. Now this enzyme is also known as citrate aconitase, right? But I am writing isomerase over here. It will be convenient if we are going to number these reactions. So the very first reaction is this: when acetyl coenzyme A and oxaloacetic acid together synthesize citrate. This is our reaction number 1. Then from citrate to isocitrate, this is our reaction number 2. Now from isocitrate, what compound is going to form? It is alpha ketoglutarate. This is our reaction number 3. Now this alpha ketoglutarate is a 5-carbon compound. So let us write over here that it's a 5-carbon compound. So as we can see that isocitrate is changed to alpha ketoglutarate. Isocitrate is a 6-carbon compound. It gets converted to a 5-carbon compound. That means it's a decarboxylation step, and 1 CO2 is being released out. Also, NADH will be reduced to NADH2. So there are 2 things happening in this step. 1, the release of carbon dioxide, so that from a 6-carbon compound, a 5-carbon compound is formed, and reduction of NAD to NADH2. This is our reaction number 3. Now let us quickly move to reaction number 4. Now from a 5-carbon compound, we will get a 4-carbon compound, and this is known as succinyl coenzyme A. So now here coenzyme A is present, so we can say that 1 coenzyme A will be added. See, there is no coenzyme A over here, but in succinyl coenzyme A there is coenzyme A. So 1 coenzyme A molecule will be added in this step, and because it is being converted from 5-carbon compound to 4-carbon, it means there is release of 1 carbon dioxide molecule, right? This reaction is also a reduction reaction. So here also NAD will reduce to NADH2, and let me just quickly write down that it's a reaction number 4. Now let us discuss the enzymes used in these steps. Let me just erase it. Now from isocitrate to alpha ketoglutarate, which enzyme is working over here? So it is very easy to remember the names of the enzymes, because this is the substrate which the enzyme is acting upon, isocitrate. So the name starts from isocitrate. You just have to add dehydrogenase. That's it. Dehydrogenase. The name of the enzyme here is dehydrogenase. Then alpha ketoglutarate gets converted to succinyl coenzyme A. The enzyme used here is alpha ketoglutarate dehydrogenase. Now, this is a 4-carbon molecule, succinyl coenzyme A. It will get converted to succinate. Succinate again is a 4-carbon compound, so no decarboxylation. Then here we can see that coenzyme A is present. Here no coenzyme A is present. So this suggests that it will now release this coenzyme A molecule, right? But in this step, what happens is a certain amount of energy is released, and how it is depicted, let us see. 1 GDP gets converted to GTP. Ultimately, this GTP is being converted to ATP, right? So, succinyl coenzyme A, that is a 4-carbon molecule, is now converted to succinate. 1 coenzyme A is released and GTP is being synthesized, which will ultimately gives us 1 ATP. Now, we have done 4 reactions. This reaction, let me name it as reaction number 5. So now, this is our reaction number 5, right? Now, from succinate, this succinate is converted to fumarate. This is our reaction number 6. Now, this particular reaction, this particular step is also a redox reaction, but the substrate that is being reduced here is not NADH. It's not NAD. The substrate here is FAD. Now here, FAD will be reduced to FADH2. So as you can see, FAD is reduced to FADH2. Now there is a difference between NADH2 and FADH2. Now NAD will reduce to NADH2 and will give 3 ATP molecules. But in case of FAD, when FAD is reduced to FADH2, it is worth only 2 molecules of ATP. So this is a very important thing to be kept in mind, that when NADH2 is being reduced, we will get 3 ATP molecules, but in case of FADH2, we will get only 2 ATP molecules. So now, let us discuss about the enzymes that are being used in reaction number 5 and reaction number 6. So in reaction number 5, now the enzyme will be succinyl coenzyme A. Again, the name of the substrate. What is being synthesized? Succinate is being synthesized, hence the name succinyl coenzyme A synthetase. This is the name of the enzyme. Then, in this step, when succinate is being converted to fumarate, let me tell you, fumarate is also a 4-carbon compound. Now, from here onwards, we will only talk about 4-carbon compounds. Succinate is 4-carbon, fumarate is 4-carbon, another, you can see malate is there, it is also 4-carbon, and then the final oxaloacetic acid, which is the acceptor of acetyl coenzyme A, is again 4-carbon compound. So now there is no decarboxylation during this chain of reactions, right? Now when succinate is being converted to fumarate, the enzyme used here is succinate. Again, the name of the substrate is coming first, dehydrogenase. This is our enzyme, succinate dehydrogenase. Then from fumarate, malate is formed. This is also a 4-carbon compound, and the enzyme used here is fumarase. I will write over here. The enzyme used over here is fumarase. Now this step, when fumarate is being converted into malate, this reaction is reaction number 7. And now malate is being converted to oxaloacetic acid. This reaction is step number 8. Now in this reaction, this is also a redox step. It's a redox reaction. Again, NAD will be converted to NADH2. And the enzyme used here is malate dehydrogenase. Dehydrogenase. So these are the enzymes. Now from malate, oxaloacetic acid is being produced, that is a 4-carbon compound, and now this is the complete cycle. So this whole complete cycle which we have now discussed is for one pyruvic acid molecule. But in case of glycolysis, we get 2 pyruvic acid molecules. So we have to multiply the total number of ATP generated from one pyruvic acid molecule with 2, so as to get the total amount of energy that we are getting from one glucose. Fine. Now it is very easy to remember the names of the enzymes. First is citrate synthetase. Why? Because the citrate is being synthesized. Next is citrate is converted to isocitrate. This is the isomeric form, so the enzyme is isomerase. Then in step number 3 and 4, substrates are isocitrate and alpha ketoglutarate. So the names of the enzymes will be starting from the respective substrates, and we will add dehydrogenase. Like in case of isocitrate, 2-alpha ketoglutarate step, the enzyme uses isocitrate dehydrogenase. Easy. Then from alpha ketoglutarate, succinyl coenzyme is being synthesized. So the substrate here is alpha ketoglutarate, hence the name alpha ketoglutarate. Then we just need to add dehydrogenase. Now from succinyl coenzyme A, we'll write the substrate as it is and add synthetase. Why? Because the succinate is being formed here. Now from succinate to fumarate. Now substrate here is succinate. So we'll write the name of the substrate and we'll add dehydrogenase over here, right? Then from fumarate to malate, we just have to replace this TE with SE, fumarase. Yeah. Then from malate to oxaloacetic acid, we'll write the name of the substrate malate and add dehydrogenase. So these are the names of the enzyme. Now I'm going to quickly discuss a small trick to remember all these steps of this Krebs cycle. So students, I'm back with this trick to remember the substrates, the steps for Krebs cycle. Now this is a simple mnemonic that I have formed. It says, can I keep selling stuff for money online? Can. The first letter is C. It stands for citrate. Then we have I for isocitrate, K for alpha ketoglutarate, S for succinyl coenzyme A, this S for succinate, this M for fumarate, this M for malate, and this O for oxaloacetic acid. Now let me just quickly jot it down for you. This is isocitrate. Now this K stands for alpha ketoglutarate. S stands for succinyl coenzyme A. This S stands for succinate. Then F stands for fumarate. M stands for malate. And O stands for oxaloacetic acid. So if you will simply remember this line, this mnemonic, as can I keep selling stuff for money online, then you will never confuse that which step has to come first, right? So this is a very simple way and it will be really helpful to you. If you find this helpful and you want more such tricks, just leave a comment in the comment section and leave the name of the topic for which you need a trick. So students, that is all for today's video. I hope you have learned the lesson, understood the topic, and I'm waiting for your comments in the comment section so that I can also have a feedback from you what else you want to learn. Until our next meeting, take care of yourself. Have a good day. Bye.